All right. Hello and welcome everyone to part 2, a conversation about patient data. My name is Casey Craig. You may know me from our support group Facebook page as Stella's mom or the SCNAA Family Book Club lady. But today I'm here serving as a Cute Syndrome Foundation volunteer and will be moderating all of the clinical trial readiness educational series. This 5-part series is aimed at helping all of us gain perspective and information as we prepare for upcoming clinical trials. The Cute Syndrome Foundation wants to ensure that our families are informed on how clinical trials work, as well as general drug development, ethics of patient data collection, and potential future therapies. This series will bring together panels of industry professionals, clinical researchers, and advocates to discuss these important details. Last week, we had the pleasure of chatting with Rachel as she shared her awesomeness forum with us. Guys, this was such a treat to celebrate our children's uniqueness, talents, and abilities. If you missed last week, no worries. We are working on uploading the recorded session for you to view at your convenience, so stay tuned. Now moving on to today's event, patient data. So patient data is incredibly important for us to move forward medical science. However, it's not as simple as it may seem, such as all the regulations and ideas of how it should be collected and who should own it and how should it be shared. It's all very complex. It's important that we know about the value of our data, our rights, and the expectations we should have about the use of our own data in general, but especially as we move forward towards clinical trials. So please feel welcome to ask questions during the Q&A session in today's event. We highly encourage them as we have some incredible panelists today to help us answer those questions. Before we get started, we have a few housekeeping items. So group interaction through questions and comments today will take place in the chat box only. So the chat box is to your right, and if you click on that, it allows you to type questions or responses for Hillary or any of our amazing panelists today. You'll also notice to your left, there's a tab titled Expo. In this area, we'll find some troubleshooting. If you're experiencing like some technical issues, you can also request some help from Megan Varner. She will be fielding those technical problems by clicking in the chat box. Before we get started, I wanted to take a moment to welcome our Duke 15Q community that are joining us as we collaborate together in this clinical readiness journey. I also want to extend a huge thank you to our sponsors for this event, Neurocrine, Praxis, and Zogenix. All right, so now I will hand over the torch to our very own Hillary Savoy. We all know her as the director and founder of the Cute Syndrome Foundation, but more importantly, she's the mom to the original Cute Esme. She truly paved the way for our kids, and I know I am forever grateful for her leadership and for her friendship. Miss Hillary Savoy.
Hi. Can everybody hear me okay? All right, wonderful. So welcome everyone. I am so happy to be here again with our SCNA Day families and pleased again to welcome our Duke 15Q community. As Casey said, I'm Hillary Savoy. I'm the founder and director of the Cute Syndrome Foundation. And we're here today for the second session in our 5-part series on clinical trial readiness. And again, the idea here is that we're really focused on creating an environment for casual conversation and learning for our community. And today we're going to focus on patient data. This is a really, really important topic as we think about clinical trial readiness because patient data plays a role not only in a clinical trial itself, but in the preparation for clinical trials. So for example, many of you may remember the survey that the Cute Syndrome Foundation conducted with Xenon Pharmaceuticals within the SCNA Day community about this time last year to help inform their clinical trial plans. And what's really interesting about that survey was that our community's really tremendous response rate for that survey was really impressive and telling in its own right about what kind of a community we are and how involved we want to be. We worked very hard on that survey to design it in a way that collected necessary data and respected the time commitments of families participating in the survey. And in that process, our team at the Cute Syndrome really learned a lot about the ethics of gathering data, the ways in which participation should be solicited, the care of sensitive patient information, especially any identifying information, and the challenges of data ownership and sharing. At the end of this session, actually, we have a bit of a treat. I'm going to share some slides that Neurocrine has shared with us that they've put together for our community to show some of the outcome of that data that's publicly available. And I think this is really important because it's one of the things that we need to see as families is that our data is being used to help understand, support, and treat our community, and it's being shared back to us in ways that we can understand and relate to. So without further ado, I want to move on to today's panelists. I have had the great fortune of learning about the power of patient data, changing regulations about data collection, shifting landscapes about who owns and should own patient data, how it's used and shared from the three panelists we have here today. And I'm really eager to have a chance to allow them to share some of their insights with our community. Really as a means to help all of you feel empowered as you weigh your decisions about involvements in interventional and non-interventional studies. So with that, I'll ask our panelists to join us as I introduce them. So today we have Dr. John Schreiber, Director of the Epilepsy Genetics Program and Associate Director of EEG at Children's National. Dr. Gabby Belfort, who is the Senior Medical Director at Praxis Precision Medicine, and Vanessa Vogel-Farley, the Executive Director at Dupe 15Q Alliance and the Data Collection Platform Lead for RareX. Welcome, guys.
Thank you.
Thank you very much.
Thank you guys for being here. I'm really excited to have this discussion today. I've been really looking forward to this. So I guess maybe where we should start is if each of you could just take a moment to explain your connection to this topic and our community. I think we can start, we can go in order. John, if you wouldn't mind starting.
Yeah, so I'm a child neurologist and an epilepsy specialist in pediatrics at Children's National in DC. I direct the epilepsy genetics clinic there. I'm director of EEG, and I've known this group now for several years. I think 2015 maybe was when I first got involved, and just been really fortunate to be involved in we have a multidisciplinary clinic for SC and 8A related epilepsy, and have done a number of projects with TCSF, some of which you all have been involved in. So thank you for your support and help and involvement in all that.
Absolutely. Thank you, John, for being always the wonderfully reliable clinician for our community. And Gabby, tell us.
Yeah, so thanks so much for having me here on this panel. I work at a biotech company in Cambridge, Massachusetts called Praxis Precision Medicine. My background is I basically been living in and around Cambridge for 25 years, going to school and working. So I did MD, PhD at Boston University, and then residency training in neurology, including some pediatric neurology. Pediatrics was at Children's, and the adult work was at the Beth Israel Deaconess Medical Center, and I've been in biotech now for almost a decade. I really feel super excited to be here talking about patient data. It's a critical topic, but I guess more importantly, I'm really excited to be connecting with the patient group today, both patient groups. I really get my inspiration for the work I do every day from speaking with patients, and it's very orienting. We push a lot of paper, and when you do that, you're like, why am I doing this? And it's incredibly valuable to me to come and speak to families and know why I'm doing it. So thank you.
Yeah, thank you for being here, Gabby. And I think it's really important for the families to understand that, Praxis has been a really wonderful partner for the 8A community. And Gabby in particular has been a tremendous resource for our community throughout the 2 years that we've been collaborating with Praxis. So thank you, Gabby. And Vanessa.
I'm Vanessa Vogel-Farley. I'm the Executive Director of the Dupe 15Q Alliance. So hi, all the Dupe 15Q families who are watching. I also serve as the Data Collection Platform Lead for RareX, which is a new data collection platform for rare disease to look at, to create more structured data around that. My journey for rare and rare data started when I was a neuroscientist. Actually started at University of Minnesota, then went to Children's Hospital in Boston for about a decade, and really fell in love with the patient advocacy side of things. Was able to spend a lot of time collecting a lot of EEGs with families, and heard about their diagnostic odyssey, and kind of wanted to figure out why they were there to give data when their kids were having a really hard time, when they were probably going to give data to something that might not affect themselves, but work more for the community, and just really wanted to bring that research side of things into the advocacy realm. So when I took the position as the executive director of the Duke 15Q Alliance, it kind of really brought together my two sides of my brain in terms of that patient advocate side and the patient engagement family side. And then on top of it, the pushing paper for all the longitudinal rare data that we've collected along the way, and putting them together, saying, when we collect data, making sure it's patient focused, making sure it's patient forward, making sure that we're taking into account the time that it takes for rare disease patients and families to fill this out, and all the other things that are going on in our lives at the same time. So I'm really excited to be here and talk about this.
Thank you so much, Vanessa. And everyone should know that Vanessa is my go-to data expert on every data question that I have. So, and I've had great fortune of working with her quite a bit for the last year. So with that, I think we should dive into some questions. I think maybe just to begin, sort of at a, setting a baseline, there's a lot of types of research that families are asked to participate in, up to and including clinical trials. And I think that boundary between interventional and non-interventional research is really important to understand. So I thought maybe, John, if you could talk a little bit about natural history studies as and other clinical research to start us off, and give us a sense of the role that a natural history study plays in research.
Yeah, great. Yeah, so thank you so much for giving me the opportunity to talk about this. It's something that is very near and dear to my heart, and something I've been spending a lot of time thinking about lately. So natural history studies are really useful for understanding a disease and getting a more comprehensive view of what happens to people that have a given disease, in this case SCN8A related epilepsy, and other types of SCN8A related neurological disorders. We get a lot of information from our bench researchers, right? The scientists that are doing the work to understand what happens when you change this voltage-gated sodium channel, what happens to neurons, what happens to other body systems. And that really helps to inform how we evaluate people, how we see people, how we treat people. But on the other hand, you as parents and caregivers and us as clinicians and seeing and trying to apply this knowledge to patients in real time, it's also useful to systematically collect information about what we're seeing in the clinic that can then also be used. It can be used for a number of reasons, and one, it can be used to better understand the pathophysiology, and to go back to the people that are doing that neuroscience or that other scientific work to try to better understand the mechanisms by which this disease has its multiple different effects. That's one. The other is it helps us to better understand the course of this illness. We know that SCN8A related epilepsy, for instance, is so much more than just seizures, right? There's all kinds of other systemic problems. Kids have issues with their GI tract, they have respiratory problems, they can have cardiac issues, and clearly developmental and behavioral problems are very common in people affected by this. And not only that, but it has effects on the family unit, I think, as this group certainly appreciates very, very well, since you're living with this every day. But the natural history study, classically, it is meant to understand what the course of a disease is over time without treatment. And the reason why that can be helpful, another reason why it can be helpful, is because it helps us to better understand the disease progression and to have a baseline when we are thinking about doing interventional studies. In some cases, the FDA can even use historical data to compare, to use these people enrolled in a natural history study as a comparator for those that are then given an intervention. Now I think that's a little easier to do for diseases that are more well defined, or that they have a more clear and consistent progression in disease. Things like spinal muscular atrophy or Duchenne's muscular dystrophy are good examples of where that kind of natural history data has been used to provide a control group for a therapy. It gets a little more complicated in something like SCN8A-related epilepsy, where it's hard to know if something like that might be possible. But it still is useful to understand the consequences and outcomes of this disease beyond just getting seizure frequency. We know it's so much more than that. So there's the classic traditional natural history study where someone comes into a clinic and then is followed longitudinally over time. It's not just one visit, but you're seen multiple times to see progression. There's all kinds of different standardized measures and things that we can administer, whether those are things like EKGs, echocardiograms like we did in the clinic, neuropsychological testing, and then just documenting other things about someone's medical history. But the registry, which a lot of you also participated in, is also a form of a natural history study. And that gives us different types of information. It gives us information on usually patient or caregiver reported outcomes. It gives us information on the genotype. What is the specific mutation or change in SCN8A that your child has, and again, how does that change over time? And the advantage of something like a registry is that the data can be collected in general in much more people. It's sort of easier to do, but again, can be followed longitudinally, can be followed over time. So all this data is really important, and as Hillary alluded to, it is something that is vital to our understanding of a disease and how we can intervene and hopefully help not just your child, and in many cases, the kind of data you're providing may not necessarily be so impactful on your child, but it may help other people who are dealing with this too.
Absolutely. Thank you, John. And I think, Gabby, maybe this is a good spot for you to fit in and kind of talk about how you take the kind of information that John's talking about and translate that into clinical trial design and some of the things that you might need to do in between, like the types of surveys that I mentioned that we did with Xenon to help you in that process.
Yeah, I think, John Lee, thank you for that, John, because you really laid an outstanding foundation. I think you think about conditions that are often studied in clinical trials, like Alzheimer's disease or schizophrenia or depression. And gosh, if you go do a PubMed search for those conditions, there's 10 to 100,000 papers on those conditions. So there's an enormous understanding of those diseases because they're prevalent. They have an extremely high incidence rate in our population. We can see what those diseases are like, and therefore, if you're thinking about designing a study, which really comes down to three things, which is who, how much of the drug, and what is the endpoint, you can begin to answer those questions just by reading on PubMed. When you have a very rare condition, there are much, much fewer papers getting into the range of vanishingly little understood. And so when John says it's critical to start to understand this patient population so that we can intervene intelligently, I think that's, music to my ears. That's exactly right. We will really struggle to put together a study that's meaningful to the families if we don't understand what the problems are. And I want to just take a small deviation here and talk to you about seizures versus the other symptoms. So for a company that's trying to develop a new medicine, we know children with SCN8A have seizures, and so that's immediately attractive because it's countable, right? And there's a precedent for recent approvals by the FDA. So we know that companies can succeed in developing medicines based on seizure frequency. So you can just immediately point to GW Pharma and Zogenix with cannabidiol and fenfluramine and say, okay, well, they picked an endpoint that was tractable. They were able to do 3-month studies and gain approval. But I think one of the big questions coming from those from that work is, to what extent do those drugs actually change the lives of those children? And so I think as we think about this community, other communities where there is developmental and epileptic encephalopathy associated with various different genes and different variants in those genes, what are we studying? Is it enough to get it to the market, or should we really be also drilling down on whether or not we have an impact on development? And impacting development is going to take more than 3 months. We know that. We know that measuring a significant change in trajectory will take a longer period of time. And so to have a longitudinal database like the one that's being collected for 8A is crucial. Then thinking forward into terms of the trial readiness work that Acute Syndrome Foundation did with Xenon, really important to, have an understanding of the set of details which can help guide those 3 decisions: who, what dose, and what's the end. And so the other last thing I'll just say is, look, there's a lot to be understood in terms of just what is the reality of interacting with these children. So when I think about SCN8A, we might have a great medicine that we've taken through healthy volunteer study. They're popping the pill through their through the mouth, in a first in human healthy volunteer setting. Then we get to the children, and it's like, oh gosh, how does this get administered through a PEG tube, or a different route of administration? Is there any risk that this medicine just sticks to the tube? So things like that are, oh gosh, we need to actually really carefully understand this as early on as possible. So when it comes time to running studies, we've hit the ground running, we've tested some things, and we're ready to go and have a better probability of technical success.
I think that's so really, I'm sure all of the parents on this session can understand that feeling of like hitting the ground with something and starting to try to administer it, and realizing that the people who designed it have no idea what it's like to actually do this on on the ground. So I think actually, Vanessa, that sort of, you guys are all great. You just queued each other right up. Vanessa,
I know.
Maybe you can talk a little bit about how the role of the advocacy community in helping meet these needs more quickly. And as Gabby said, we're dealing with very small patient populations. We can't afford to only capture 1% for the clinical trial. We have to, for when we're looking at studies, we need to capture everybody. And how can the community situate themselves to be helping? And how is that, the ideas about that change? How are ideas about that changing?
I think that ideas around data and data collection are changing quickly, especially in the rare disease world. Natural history studies are awesome. They're kind of the gold standard of the description. It's kind of like all the physicians that see, like they're called like the centers of excellence. So we have DUP15Q research network clinics. And what you do is like one person sees the majority of DUP15Q patients. A natural history study essentially makes encyclopedias what's in their brain, what they see clinically, and puts it onto paper, makes it Googleable. So the next person that's diagnosed with this, like they go into the neurologist, they're like, I have no idea what CN8A is, or I don't know what DUP15Q is. They can Google it and find these natural history studies, find these data sources that are coming out of clinic. So these are clinical reported things. It's a physician like John saying, like, this is what I'm seeing in clinic. These are kind of the endpoints that I would recommend, clinically we could evaluate over time in response to treatment, whatever that is. But the portion that Gabby was talking about, that whole other thing of what the community needs, is a huge, huge area that is really ramping up. It's these registry questions. It's somewhat like registries right now are such a misnomer in this space, because registries back in the day were like contact registries. Where do people exist? Where they live, and maybe some ages. Right now, these registries that are patient reported are becoming natural history studies. These are the groups of patients who can't get to see John at Children's National to be in part of the natural history study, but they're able to report on what they're seeing at home. And these kinds of things put together become a wealth of information for biopharma partners who are developing treatments. It's able to say that most of the kids have PEG tubes, most of them don't. Or when you're looking at the way of administration, it might not work so well for one population, but it might work better for another. I know we were recently part of a clinical trial. We were talking about the administration that you couldn't smash up the pills. So you couldn't hide them in anything, but swallowing was a problem for our kids. So working around those things when you are partnered with biopharma is really, really great to make sure that the community has a voice in that. So when you're talking about protocol design, or you're talking about ways of administration, that's their job. That's Gabby's job. That's his job to go back to the formulators and say, we need a different route of administration. We need a different way to make sure that it gets into these kids and stays in these kids and over time. So I think that piece, those things coming together, where if there's a survey coming out that is well designed, that's able to get at what your community actually is made up of, what your community needs are, is really, really important along with these natural history studies. Because what happens is if you can get that parallel data where it's parent report data that's really, it has a lot of fidelity to what John's seeing in clinic, that makes the ability to put a treatment into any population really, really important. We're doing the same thing in the Duke 15Q area, where we have our patient reported data and we have our clinic reported data. We want them to mirror each other. We want them to be traveling together, because that becomes a much, much more effective community partner for biopharma and for other different interventions than ever before. And these are huge advances in this space. So I think that you as parents and you as patient families, who are really the ones providing the data, whether it be in person or online, making sure you understand the reason that the survey is coming to you. Where is it coming from? Who designed the questions? So are they questions that are actually designed to get at the answers that you want to give? Or are they questions just kind of like you'd see on Facebook polls? Those are two very different things. So making sure you know where it's coming from, know where your data is going, meaning that you want it to be shared in a way that's secure, that it's getting legs, essentially, that it wants to be shared amongst people, because not just one biopharma is usually interested in this space. You want to make sure that you don't have to keep answering the same questions over and over again, because data can be shared, and making sure you're able to understand where that data is going. So those are a couple ways in terms of these are really big changes in the rare disease space, especially around data, and getting involved in making sure that you're very, very educated about what you're enrolling in is important.
And it seems like that's so important, Vanessa, that, especially as these communities are growing, that we need to be able to quickly respond to the things that we're seeing in the community alongside with partners like Gabby. So to be able to develop questions that we need answered quickly and turn that around quickly as a community. So I guess we've established it's really important to share our data. So I guess the flip side of that is, what types of protections are in place for our data, or should we expect to have in place for our data? What kinds of things should we look for to, make sure that rules are being followed and that we are comfortable sharing our data?
Ooh, we can talk about governance super quick. I nerd out about governance all the time for data. It's one of those things that we see a lot of surveys that come out. I think all of us have amazing social media communities for our groups. Those are wonderful starts. It's where you can get these ideas that you see, where you can kind of see trends and that sort of thing that are kind of bubbling up in your community. Right now, especially as your community grows, meaning they actually get older, lots of good things kind of come out that you might not have known about the natural history before. And then people are like, well, does your kid do this, or does your adult do this? All of those things are great. But to be able to capture that on paper in an analyzable way is an art. And when you create surveys and when you create the registries, or when you create natural history studies, you want to make sure that the way that that's created and what you're consenting into is actually answering those questions. It's getting that data in a way that's secure, that the survey is actually created in a way that the data can be analyzed, and that where it's going to be shared to. So if it's, a lot of the times the transparency is that if it's a biopharma partner who's sponsoring the survey, it's very clear that that's who's going to be getting the data, and at the same time, what data they're not going to get. So they're not going to get your name, they're not going to get your child's name, and that sort of thing. They're not going to, Gabby's not going to knock on your door and be like, hey, I saw your data here, and because that's not good. He should not be able to find you unless there's something that's specific that they say, based on your data, I've went through the community, and it should be Hillary or whoever has that data stewardship in that governance area for your data that can knock on your door and say, or your physician, that John has been identified through this clinical trial that he's got a patient that has great data that would be a great opportunity for this, then he's the one, because he's educated in the interaction, he's educated in and covered with all the HIPAA guidance that they needs to be in place, and he knows how to answer questions on both sides, because you want to know the person's coming at you that they don't have an angle. They're able to say answer the pros and the cons on both sides.
And I think maybe Gabby, you could speak a bit to why, above and beyond, I think we're familiar with the protections that are in place for health information, like when it comes to our physicians, but when we're dealing with a clinical trial, the rules have to be followed for other reasons in addition to our personal health information.
Yeah, that's right. So, I think I'll just jump in and say the way trials are typically conducted is for the participants to interact with generally a clinician at one of the major medical centers. And so it's actually not that different. I think that the key thing is that sponsors like the company I work for that are running clinical trials typically are very cautious not to ask for this information because it's not ours, really. You should really be protecting your privacy, and you can Google the HIPAA, or you could Google what PHI is, personal health information, and you really shouldn't be sharing that kind of information. I think there are very good reasons to protect your privacy, and certainly I'm not here to say that they shouldn't exist. They should, and they're important, but they do pose a little bit of a challenge in terms of recruiting for trials, right? And so I think what Vanessa was saying is absolutely correct, that it's the job of the patient advocacy group as well as the clinician that you see to be aware of ongoing trials so that you could become enrolled in them when they come down the road. And so I do think in terms of privacy, there are some very clear instructions for industry partners where we're not supposed to go. So I'm not supposed to know any of your names, or I can know what state you're in, but nothing smaller than that. I'm not allowed to know town or village or anything like that. Social security number, email addresses. Those are all examples of the 18 different subsections of what is called PHI, or personal health information. It's absolutely worthwhile to educate yourselves on what that is and why you wouldn't share that in a survey or in a trial.
And I just put up the link in the chat to the PHI list that you sent along, Gabby. And I think it's important for families to think about that, and in relation to, again, any study that they're involved in, that they shouldn't be identifiable, and that they have, I guess the next step is sort of like what questions should they be thinking about before they make a choice to be involved in any study. Based on what's actionable, what other things should they have in mind?
I was going to add to that.
Make sure that you're asking questions of. When you're talking about questions, make sure you're asking questions of your physicians and the people who know most about the trial. Just as an FYI, we have great social media contacts, but always default to people who know the most about it. The person at the biopharma partner, your physician, and that sort of thing. Other parents are going to have a lot of opinions about it and maybe have some information, but always default to your physician. So sorry, I didn't mean to interrupt you.
No, not at all. And anytime you are considering a study like that, so there's just in general, it's one way to keep apprised of clinical trials is clinicaltrials.gov. That's another great website to follow any clinical trials that are going on, or natural history studies and similar kinds of things. But as Vanessa and Gabby were saying, PHI, anytime you're collecting PHI on a research basis, at least at our institution, we have an IRB, we have an institutional review board that is going to look at what are the risks and benefits. And as Vanessa was saying, it's important to not just look at what's the safety, what are the risks you might experience, what are the potential benefits that you might gain from this, but also looking at data validity. So the IRB really looks to say, what are you going to do with this data? Just as Vanessa was saying, it's not enough to just collect the data. You have to actually have a plan to handle it statistically, how to deal with it, and make sure you're collecting valid, appropriate data. that's going to be helpful and going to answer the question that you pose as part of that research study. As part of any research study, you're going to go through it. So those of you who have gone through it know, but you go through an informed consent process. And that person who's doing the informed consent, usually the investigator on the study, is going to talk to you about all those things, about what the risks are, potential benefits, and also what are the alternatives. At every part of an informed consent is, what are the alternatives to treatment? And if there are things out there that might be more appropriate for your child, let's say it's an interventional trial, then that's the onus is on the investigator to make sure that you're aware of those things and to have that discussion with you.
And I think, John, that's really important to discuss the IRB piece, because that's something that not everybody is fully aware of. And it's something, for instance, in designing the survey with Xenon, we had to go through an independent IRB. That IRB approved all of the outreach language, all of the, everything that we said to families was approved through the IRB. They take those things very seriously. And also, not all IRBs follow the same protocols. There are different institutional IRBs, there are independent IRBs, and then also not all jurisdictions are the same. So, for instance, with the Xenon survey, we could only do outreach to Australia, US, UK, and Canada, because we were only following the rules associated with those jurisdictions. Right. We couldn't collect data in Europe because the rules are different. So
super interesting. I guess I'd just add one thing. I think we're talking a lot about protecting people. I think I would just add one other thing, which is if you're gonna give data, shouldn't you get something back? Like some perspective on what was collected and what was learned about the community. And so I just would really encourage you, if you're gonna participate in a natural history study or a registry, that there's some kind of insight into it. And a second but related topic is, I've heard this question a number of times now. I don't have a good answer for it, but I think it's something we should talk about, which is if you give your data in a study, we'll call that study X, and then down the road somebody wants to run study Y, why can't that data be merged into the next study? How can we collect data or aggregate data from different studies that have been collected, maybe in different ways, but could be interesting when pooled? Are there ways of doing that? And then that's really where it gets tricky, because somehow we need to know that you've participated in more than one study. How are we going to do that without violating this personal health information? So there are questions around this, and we're still advancing how to do this just the right way. But for me, it makes good sense, if you've participated in multiple studies, to be able to aggregate that data.
And I think Vanessa might be the person to speak to that.
So it's called federating data. It's a big push right now in the rare disease space, especially since I have a colleague of mine. She has a daughter with a rare genetic condition, and she's participated in no less than 20 studies, including 2 clinical trials. So meaning in all of those 20 studies, she has probably filled out 20 times the same questions over and over for her child. So if you combine those data sets and you connected them, she would have an amazing longitudinal data set, but she can't. And so what the big push around rare, and actually this is what RareX is looking to do, and that's one of our big pushes, is that if you have data that exists on your patients in different places, it could be at a biopharma partner who's willing to share in a linked way through technology, or if you were part of a research study, being able to have access to the data. And when you sign a consent form, you should always look at that consent form to see that it says, I'm able to share my data, or this data will be able to be shared and accessed later by people outside of this. Oftentimes in biopharma, it's totally fine. There's embargo periods, meaning there's time periods that they won't connect until either something's to market or they've taken it off the shelf and it's not ready to go. But for the natural history studies that people participate in, oftentimes it's just, there's sometimes a technology thing. So the big push is when you're reading through those things, making sure and talking to the investigator and saying, is there ways that this data will be available to other people? If it's tomorrow or if it's a year down the path, you should be able to say that. And also, if I'm interested, if I participate in something else and I want to connect that data, as Gabby was saying, can I come back to you in a year and say, I'm participating in this other thing? I might, you know, can you transfer some of that data or connect it up somehow to this researcher? It'd be super helpful. It's one of those things, what am I getting back for this 8 hours of time I'm spending is really, really important. There's also, sometimes it could be that they just can't. And there's different reasons for different things on top of it. So there are definitely times, but making sure you're having that conversation when you're being consented. Human subject protection is really, really big on this because IRBs want your questions to be answered. They want the human, you're the human that's providing the data. They want your rights and your thoughts to be answered on top of this. So in terms of data federation, especially as you're going forward, making sure that you're having that conversation saying, you know, is there a possibility that this can be shared? The good part is technology is actually the answer to a lot of these things where you can federate the data in the background. It might not be returned directly to you as the patient because of different, again, these different human subjects protection things, but it can be connected to your data on the background and being able to be analyzed together by a researcher who has a distinct research question or something that they're able to get those statistics that John was talking about before, that they're looking at the validation of like some statistical question. So lots of good things happening in space, lots of good conversations happening in space. And it's really, really exciting. But you guys as community members and hopefully some researchers who are on here, making sure that you're open to that idea of data sharing. It doesn't have to be identified data, but making sure that linkage can happen. So when you talk about there's a code that can travel with you and it can connect those codes. So.
And I think that's so helpful and so exciting that this is, you know, a moving target in the community and that there are people working on this. And I think it's also, I'd like to ask Gabby for you to talk a bit. In my experience with biotech partners, I would say that you guys have had a much more open approach than most. And I'd like maybe to see if you have some comments on how and why that is a strategy that you guys have taken.
Yeah, I mean, I don't know if it's a strategy or an ethical approach, right? I mean, my genuine feeling is if it doesn't involve one of our compounds and it's just natural history data, then we should be willing to give it to any other company to try to help the community. Like we do have an ethical obligation as we collect data to make it available so that other companies and other approaches can benefit from it. I mean, I think John at the beginning was talking about probably Avexis utilizing data from the Biogen effort to develop their medicine for spinal muscular atrophy. So there was quite a lot of natural history data available for SMA, both from the Biogen work, but also from external natural history work. And then they were able to use that as an external control in the development of Zolgensma. So that's the kind of really heartwarming type of thing that I think we should be really compelled to do. I understand that if a company has their molecule involved, then if it's, you know, early on and difficult to interpret, they may want to collect more data before they are sharing that. But, you know, if there's no molecule or therapeutic involved, there should really be no competitive reason to keep that data secret.
I think the research world in general is moving towards this approach anyway, just recognizing the need for more of a collaborative effort. Rare diseases, and you know, in that sense, I think you find less and less people that are doing things within their own silo, and you know, reaching out to others that are doing work in the space. And that's one great thing about working in this community is just the level of collaboration is something I haven't ever experienced or seen before, and everyone has been very open to, you know, to sharing data. And you know, to speak to what Vanessa and Gabby have been saying, I mean, we know it can be a huge commitment for you all to share this with us, and it's not something we want to take lightly, and we want to take full advantage of all that data that we can.
And I think that it's also important, John, to highlight what happens across the advocacy community and the parent community and clinicians as well. I think you're right that the 8A research community is spectacularly good at sharing, and we are very, very fortunate to have benefited as a community tremendously from that. And one of the other pieces of that is that, you know, John and some of the work that we've been doing about sharing observations within the family community and trying to design studies and interventions and ask questions together as clinicians and advocates. And I think that that's another spot for important collaboration around patient data that's more anecdotal data, but it's also important data.
Yeah, I mean, there's questions that will come up from one clinician or a parent or something, and then we like to be able to ask the group. If we ask the experts first, the clinicians that are doing this, we'll ask Hillary. And if no one's seen something, it may just be something that hasn't been reported. I think a great example of this is patients who have undergone a hemispherectomy, removing a hemisphere for epilepsy surgery. Shouldn't apply to this group, hopefully. But it was identified that I think around 10% of those kids could develop late onset hydrocephalus, and that was all done through work with the caregiver advocacy group. That was found.
It's insane to see, even though I've only been in the rare disease world for just under 5 years now, and even when I started in it to now, the opportunity to put the power in the patient's hands is really, really important. To bring those questions forward, where doctors are listening much more often, biopharma partners are listening much more often to what the patients are seeing, and creating data collection efforts around that. So like the Brain Recovery Project, I think was the Brain Recovery Project who did the study for the hemispherectomy, wasn't it? With Monica?
I believe so. I know Adam Hartman also, I think he was involved in that too.
Yep. And so it's one of those things that when you bring the community bringing things forward in a way that you can collect data really effectively, it's so powerful. Additionally, the time, like the Zolgensma idea or the Zolgensma indication is insane. They decreased the time to market for that drug, the life-saving drug for muscular SMA, by half or even less than half. Just by being able to partner around data, by listening to what was happening to the people who were in the treatment, they changed the outcome measures during the trial, which is almost unheard of, by working with the FDA, working with the biopharma partners, working with the PIs at the sites, and listening to the data that was coming from the patients and the patient groups and the physicians. And it was amazing. And we are all learning from that piece, that we are changing the way that we're changing the paradigms through people like this, through organizations like Gabby's, who are willing to do that and give the data back and really, or give the data up to other places to use it more effectively. Because in all of these, time is brain. Like when we're talking about neurodevelopmental disorders and we're talking about we can't wait the 10 to 15 years that it used to take. We can't. Time is brain in all of these, and we need to make sure that that's heard. I'm preaching to the choir, obviously, but I think for the parents to really hear that there is a change. The winds of change are being felt all over the place, and that's, you're looking at some really great champions in that space.
Wonderful. Thank you. I do want to be sure that we leave some questions, time for questions from families. I see we have one queued up. If others want to add to that. Meanwhile, I just want to quickly take a moment to show you guys the Neurocrine slides. I think it's really important to end the conversation on actually looking at something that has come out of us providing our data and recognizing that our data is valuable. And we need to be, as Gabby said, we need to be asking, what are we getting back? So this is very high level information at this point, and I'm going to go through it very quickly because I'll share it on the support group for
Hillary. Can I just say one thing just before you go
Absolutely.
there, which is there was one thing that we didn't hit, which I think is critical.
Oh, go for it.
A lot of rare disease groups are talking about developing specific endpoints for their community. You can't do that without data. So, like, just to be clear, if you think that, that the standard Vineland and the Bailey aren't really capturing your kid and there needs to be something specific, well, then how are you going to develop that without really a ton of information about these children? And, I want to say, like, there's a lot of reason to believe that developing those endpoints that can better measure changes in your children has value. So, and just to add that on top of all of the other reasons we mentioned, conducting trials, but like, I said endpoints are important, but creating your own specific endpoint, gosh, that's sort of maybe the gold standard or the optimal outcome.
And it's interesting that you mentioned that, Gabby, because Vanessa and I are both on the coordinating committee for the Rare Epilepsy Network, and we're doing a lot of work on this across the DEEs, and trying to look at what the community needs, and it's absolutely essential. And you're right, we can't do it unless we have the data, and we have good data. Okay, so I'm going to just do this quickly because I want to be sure to give time. I think you guys can see the slides. So this is just some background information. It was a 36 question survey, 125 responses with 116 qualified responses. Seizures, most common type was generalized tonic-clonic. Most common comorbidities, intellectual disability and hypotonia. Most patients had taken but stopped at least one or more prior treatments. We have information on age at diagnosis and seizure onset. Average patient age in the survey was 9 years. 96% of patients had a genetic diagnosis of SCN8A DEE or SCN8A related epilepsy. This is the country breakdown. As I mentioned, we were limited in the regions that we could conduct the survey. These are the comorbidity conditions: intellectual disability, hypotonia, movement disorder, difficulty swallowing, G-tube, and so on. And then I think the families will be interested to know seizure frequency. Over 40% of patients reported having multiple seizures a day at onset. And after diagnosis, over 20% of current patients reported continuing to have multiple seizures a day. And this is really important, I think, for families to understand where they fit in the picture and how their kids are doing. And then, you know, a note that this is an ongoing analysis. Both John and I are working to help them analyze the data to understand patterns, discontinuations and usage patterns between specific treatment classes and effect on patient-reported seizure control and quality of life. And these will be shared in at our December meeting as well. So hopefully in much more detail. So I wanted to be sure to show you that, and we will share absolutely in the support group. So we do have a few questions that we've only got about 6 minutes left because these discussions are always so great, and we could go on and on for ages. But I want to be sure that we hit a few of these. So Mackenzie asks, how can we as parents assure that that happens, I guess, meaning getting something back from giving our data so that our hard work doesn't just sit somewhere? And is there a risk from pulling information from a study we start to feel uneasy about?
So I'll start off, and yeah, just to say that I think it was mentioned earlier by Vanessa that folks should read carefully these consent forms. Ensure that they have content in them that says that you will be getting something back. And if it's not there, then it's something really to bring up to the people conducting the study that it's crucial that you, in order to consent, you're going to need to be able to get something back. So that's the simple part, is to say that. I think I wanted to just take a second and acknowledge that these consent forms can be 20 pages long, super complicated, and overwhelming to me, to everyone. And so don't feel that you have to sign these things immediately on your first view. Take them back, bug Hillary with them, come to Vanessa, talk to John, Casey, me, find somebody who can help look through it with you and help you understand the terminology and the conditions for participating in the study. Take your time.
If you're looking for something specific, the person who's giving you that consent probably has had a love-hate relationship with that consent since they wrote it or had to put it together. And say, can you show me the data sharing parts of this? Can you show me these areas? Because they should be able to flip through the 75-page consent and be like, here, this is what you're consenting for. Because believe me, each individual word on that page they've looked at, had to go over, looked at grade level, all those things. They should be able to answer your questions and point to where it is in the consent form. If they can't, they're not a good researcher. But it should be able to do that. But again, bring it back, ask questions before you sign it, really understand what it's doing. But they should be able to say, are you able to share it? Where's this going to go? And all of those other things.
Yeah, and I think that's really important, Vanessa. I think a lot of our families have gotten used to the idea that you can ask your clinician a bunch of questions that maybe before they had their child, they never in a million years would have questioned their clinician in that way. But once you have a very fragile child, you start, I'm sure, John, you've been on the receiving end of a lot of very difficult questions and challenges, right? And we start to get comfortable with that notion. And the same is true of research, right? We have the right to ask questions about how our data is being used. And as you say, if they can't answer the questions, that may suggest another problem. Okay, so we have another question here from Chinzia. How can we as parents assure that the data collected are clinically usable? Also, if they need to be joined with other data sets. Sometimes research groups collect data in different ways that they can't be combined.
Yeah, I can try to tackle that one somewhat. I mean, so, and I think Vanessa got into this some. It really gets to the data sharing plan, and if one exists, and sometimes things can actually change after the fact. So you can always modify an IRB. You can go back and reconsent subjects potentially, and sometimes the consent form will actually include, whether or not they can. Again, for participants in other kinds of trials and that sort of thing. So again, it does get back somewhat to the consent form. As far as if the data collected is going to be valid or not, or useful, there are a few checks for that, and one is the IRB, one is the the person who's running the study. Hopefully, if you are enrolling in this trial, it's something where those questions have been properly vetted. But in some cases, it's, Gabby got into this a bit, the data that we need to collect to form some kind of a, complete, more comprehensive outcome scale, or, whatever that, it's a bit hard to know which of those things will be useful. So in all likelihood, not everything will be useful. And I'll tell you, in collecting data, just if you look at neuropsychological outcomes, for kids with the various kinds of developmental and epileptic encephalopathies, it's really challenging, in part because there's what we call the phenotypic heterogeneity. Kids can look so different, not just with seizures, but with their developmental outcomes, that there's not necessarily one tool out there that's really going to capture the range. There's a floor and a ceiling effect for some of these things that just may not be able to fully capture how a child is doing in that regard. So, depending on the study, there may be some data collected that ends up not being useful, but it is a bit hard to know beforehand what that's going to be. You have to look at the hypothesis, the question they're trying to ask, and if it seems like it could potentially be valid.
Additionally, just for retrospective data stuff, in terms of connecting data, harmonization is a thing that people tend to do, that similar questions can be compared by asking a third question of them. So there's ways around, there's ways to look at data that way. Also, if it's not necessarily like it was collected in different ways, it could also inform future data collection, as Jen was saying. And so it's really important that if it exists, especially in this rare space, to see, to as much as possible share. So. Yeah,
I was going to say everyone should vote. And like some people live in states where you may think your vote's not going to be important because the polls show that it's going to go one way. Well, sometimes they don't. So more data is good.
I like you combine that with a get out the vote effort.
Vote.
Vote.
Well, I think we have come to time, and I want to be respectful of everyone's time today. But I am just so grateful for this conversation, and for the three of you for showing up and engaging with our community. And Gabby has already offered all of you as contacts now to our patient community. So I can speak for the 8A community not being shy. So you may be receiving some emails, everyone, with questions. But thank you guys again, and for helping inform us on this really important issue. And thank you, Casey, for moderating today. And thank you, Megan, for, as always, being our media extraordinaire volunteer and hiding in the background, but we have to acknowledge her, even though her face isn't up here. And thank you for the 8A community, to the 8A community and the Dupe 15Q community for showing up, because it's fun to have these conversations, but it's even better to have them when there's an audience. And also a big shout out to our sponsors, again, Neurocrine, Praxis, and Zogenix for supporting this event. We will see you next week and be talking about Clinical Trials 101 with some other really exciting and interesting guests. So thanks, everyone, and look forward to seeing everyone next week.
Thank you.
Thank you, guys.
Thank you. This was fun.
Thank you.